Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on recombinant DNA. In our last lesson, we discussed how bacteria are used in sewage treatment applications. We discussed two methods in particular: percolating filter method and activated sewage method. Today, we will be studying the role of bacteria in recombinant DNA work. We will look at how foreign genes are inserted in bacterial plasmids and how bacteria are used as vectors in biotechnology. Are you ready to begin the lesson, students? Based on what we have learned in our previous grades, what do you know about genetic engineering? Discuss this term with a partner or in groups of 3. You may begin.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, everyone. I am sure you had some interesting information to share with one another. Genetic engineering is the practice of transferring genes from one organism to another organism. The organisms can either belong to the same species or they can be from different species. The organism that receives the gene becomes a transgenic organism, or a genetically modified organism. As we have learned, genes are sections of DNA within an organism. They supply code for particular proteins. You may be wondering how this applies to bacteria. If a gene is successfully transferred from an organism into a bacterium, the genetically modified bacterium is capable of making a protein that its modified gene codes for. In other words, bacterium can be altered for different characteristics or for specific purposes. Some examples of ways modified bacteria are used are in antibiotic resistance, medicines, human growth hormone, or human insulin. The development of 3 techniques made genetic engineering possible. The first was that genes can be cut out of a DNA molecule using enzymes called restriction endonucleases. The second was that genes can be inserted or tied into another DNA molecule using a ligase enzyme. The third technique was that genes transferred into other cells using vectors. Vectors are a means of transferring something. Vectors are either plasmids or viruses. Plasmids are small pieces of circular DNA that are found in bacteria. Let us now go through the stages of genetic engineering. First, plasmids are isolated from a bacterium. The plasmids are opened with a specific restriction endonuclease enzyme. The gene to be transferred is cut from the donor DNA using the same restriction endonuclease enzyme. The opened up plasmids and the isolated gene are mixed with a DNA ligase enzyme to create recombinant plasmids. Bacteria are incubated with the recombinant plasmids. Some bacteria will take up the plasmids. The bacteria that have taken up the plasmid now contain the gene from the donor cell. This could be a gene controlling the production of human insulin. Now, we will look at how bacteria are used to make recombinant DNA. Bacteria are incubated with the recombinant DNA. Some bacteria will take up the plasmids. The bacteria that have taken up the plasmids now contain the DNA from the donor cell. The DNA will replicate, and the bacteria will develop and grow. Once the bacteria start to grow, every new bacterium will contain the recombinant DNA. Students, let us do an activity to review what we just learned. You will see a diagram on screen of the stages of genetic engineering using bacterial plasmids. Take a minute or so to quickly draw the diagram on a sheet of notebook paper, and then label the stages. You may begin now.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, students. How did you do? This is what your diagram should look like. Your diagram should include the following steps. First, the plasmids are isolated from the bacterium. The plasmids are opened with a restriction endonucleases enzyme. Next, the opened plasmids are connected with a strand of DNA that has been cut from a donor. The DNA strand and the DNA molecule are connected using a ligase enzyme. Recombinant DNA is formed. The recombinant DNA plasmids are incubated with bacteria. Some bacteria take up the recombinant DNA plasmids. The bacteria that have taken up the plasmids contain the DNA from the donor cell and will grow and develop with this new DNA. Were you able to name all of the stages? There are quite a few, so it is important to review this process again with teacher. This brings us to the end of our lesson. Today, we learned about recombinant DNA. We learned the role of bacteria is producing recombinant DNA, and we studied the stages of this process. Until next time, thank you, teacher. Thank you, students.